Hello, everyone, and welcome back. I know this was a very short break to today's session on lysosomal pathophysiology. I am Federico Mingozzi, and I'm very happy to be the moderator of this exciting session. So today's first speaker, the keynote speaker, is Carmine Settembre. He's from the Telethon Institute of Genetics and Medicine in Pozzuoli and the Federico II University in Naples, Italy. And the title of his talk, you can see it on the screen, is Phagy, Function, Regulation, and Implication in Lysosomal Storage Disorders. Carmine, is your turn to go. Thank you.
Okay, thank you, Federico, for the kind introduction and invitation. So welcome everybody, good morning or good afternoon. Today I will show you some data that we have generated in the lab in the last years, and then especially at the end of my talk I will try to convince you that those data might be important for understanding the pathogenesis of lysosomal storage disorder, including MPS. And so eventually to develop new therapeutic options, especially for the features that are still unresolved with the current therapies. So we all know that the lysosome is the main degradative organelle of the cell, and it is able to digest virtually any biological material thanks to the presence of lysosomal hydrolysis, which are within its lumen. And those enzymes are basically enzymes that are involved in the step-by-step degradation of complex molecules in more fundamental units that can be then recycled back from the lysosome to the cytoplasm, and then the cells can actually use these elementary units as a building block for de novo complex molecule or as a source of energy. And this is particularly important during lack of nutrients, supply of nutrients from extracellular sources. So substrates can be delivered to the lysosome via multiple apparently independent pathways, such as endocytosis, which is the pathway that is involved in the delivery of plasma membrane and extracellular substrates to the lysosome, while intracellular material are delivered to the lysosome largely via autophagy. Currently, we know three different types of autophagy: macroautophagy, microautophagy, and chaperone-mediated autophagy. So today I will share with you data about macroautophagy, which is considered the most important also from, at least from quantitative point of view, for the delivery of substrate to the lysosome. So this process starts, the macroautophagy process starts with the formation of a new vesicle, which is known as autophagosome. And so this autophagosome has the capacity to sequester intracellular material, such as organelle, large protein complexes, and also invading bacteria and viruses, and then deliver these substrates to the lysosome for degradation. And this is happening through a step that is the fusion between an autophagosome with the lysosome. And so upon the fusion, this material is delivered within the lysosome, recycled in smaller molecules, and then these molecules can be recycled back within the cytoplasm. So currently, we believe that there are two different types of autophagy. One is a bulk unspecific autophagy, which is triggered by lack of extracellular nutrients, intracellular nutrients, nutrient supply from the extracellular source. And so this drop in the nutrient inhibits the mTOR kinase. And so the inhibition of the mTOR kinase triggered the activation of several proteins involved in the autophagosome biogenesis. And so autophagosomes start to form very efficiently within the cells, sequester through mostly in what is considered an aspecific mechanism, several substrates, and then deliver them to the lysosome for their degradation. But more recently, a new type of autophagy has been described, and is called selective autophagy. So this type of autophagy seems to occur in a manner that is independent of nutrients level within the cells, and is instead triggered by the cargo itself. So if you have a damaged cargo within the cells, such as a damaged mitochondria or a polyubiquitinated protein aggregates, this cargo is itself able to trigger the biogenesis of an autophagosome, because it can actually promote the arrival of autophagy proteins surrounding the cargo, and then these autophagy proteins start to build an autophagosome that sequesters exclusively this cargo and target them to the lysosome for degradation. And this, of course, you can imagine as a very important quality control function for the cells, because it's implicated in the removal of aged or damaged material from the cells in a manner that is independent of the nutrient status of the cell. So key players in this mechanism are the so-called selective autophagy receptors. So this selective autophagy receptor has the ability to bind to cargo that needs to be degraded, and on the other side has the ability to recruit autophagy proteins, such as LC3, which is a protein that is associated to the autophagic membrane, but also other proteins that are important for the biogenesis of the autophagosome. So this cargo really play a key role in selective autophagy, because from one side, these receptors really play a key role in selective autophagy, because from one side recognize the cargo that needs to be degraded, and on the other side help the recruitment of autophagy protein that make an autophagosome surrounding the cargo. So in my lab, we study a selective type of autophagy that is involved in the degradation of endoplasmic reticulum. This type of selective autophagy is known as Phagy, which is a receptor-mediated degradation of fragments by lysosome. So this Phagy starts with the formation of an vesicles, and these vesicles can actually be sequestered within an autophagosome and then delivered to the lysosome for degradation. This is actually helped by the so-called Phagy receptors, so which are proteins that possess in the cytosolic domain the so-called Lear domain. So the Lear domain is a stretch of amino acid that allow these proteins to interact with the LC3 proteins on forming autophagosome membrane. And so in this way, these vesicles of is actually can actually be recognized by a forming autophagosome. So these autophagy receptors, these Phagy receptors in particular, we study one of the first Phagy receptor identified in mammals that is known as a FAM134B. So FAM134B has a membrane transmembrane domain that is important for promoting membrane curvature, and then as a Lear domain that is important for the interaction with the LC3 protein. And so in this way, it not only helps to reshape the endoplasmic reticulum in order to form a shape that can be allocated within the autophagosome, but also can physically interact with autophagy proteins, allowing the incorporation of these fragments within an autophagosome. So Phagy is currently considered important for membrane turnover, the regulation of sites during starvation, recovery from stress, and I will show you some data that we generated in the lab suggesting that Phagy has an important quality control. So those are published data, so I will be very brief on this part. in order to study the role of Phagy, we generated cells lacking the Phagy receptor FAM134B, and then we did the proteomic analysis in order to understand whether there were cargos that were actually accumulating in the absence of Phagy. And we found that collagen were actually the most abundant cargo that accumulate in cells lacking the Phagy receptor FAM134B. And this was confirmed then by Western blot analysis and by immunofluorescence analysis. So we really get interested in this data because, as you may know, collagen are very important proteins. So they are the most abundant protein in animals. They are the most prevalent component of our extracellular matrix. And we know that collagen are very large protein and very complicated to be folded. And so this is a really a demanding process for the endoplasmic reticulum. And it's also associated to several diseases. So to date, more than thousands of mutations have been identifying collagen genes and are associated with several human disorders, such as osteogenesis imperfecta, chondrodysplasia, hairless Downers, and so on. So really, we thought that our finding in cells with deficiency for Phagy might be important for pathophysiological reasons. So we basically demonstrated that in a normal cells, collagen, a fraction of collagen that we consider to be misfolded is delivered to the lysosome for their degradation. And you can appreciate that because if you treat cells with baphyllomycin, so baphyllomycin is an inhibitor of lysosomal acidification, you can actually see that collagen accumulate within the lumen of the lysosome, that are these green rings, and collagen is in red. But if you block autophagy or if you block Phagy, you can actually appreciate that there is no more collagen delivery within this lysosome. So suggesting that the Phagy is actually important is the mechanism by which collagen is delivered to the lysosome for their degradation. And in addition to that, we also found that the calnexin chaperones, which is an important protein which is an important chaperone that is involved in collagen in the folding of many proteins, including collagen, is actually also implicated in the recognition of collagen by the Phagy receptors, and then it's involved in the delivery of collagen to the lysosome. So in conclusion of this first part, we basically demonstrated that Phagy has an quality control function. And among the different clients, clients that can be delivered to the lysosome via Phagy, we found that collagens are indeed notable substrates. And we also found that the receptor FAM134B is indeed forming a complex with transmembrane protein calnexin. And this complex is actually important from one side to binds the misfolded procollagen, and on the other side to deliver this misfolded procollagen to the autophagy machinery for degradation. So in this way, autophagy is preventing the accumulation of non-properly folded proteins such as collagen within the lysosome, within the endoplasmic reticulum. So it's preventing the accumulation of this misfolded cargo within the endoplasmic reticulum, which we know are causing cellular dysfunction. So we also demonstrated that we also identify a signaling pathway how Phagy is regulated. And this study started from an observation that was made in my lab a few years ago. So we found that in chondrocytes, which are the a main regulator of bone elongation and cartilage formation, in chondrocyte, the inhibition of autophagy was actually associated with an dysfunction. And we also found by an high-content screening approach that FGF signaling in chondrocyte is a major regulator of autophagy. So this led us to propose the hypothesis that maybe the FGF signaling in chondrocyte was indeed controlling Phagy. And so we did different experiments which support this hypothesis. I'm just showing you some of them. So as you can see in these cells, by treating chondrocyte with FGF18, you can actually appreciate that ER fragments, which are now in green because they are staining with an ER membrane protein CLIMP63, ER fragment accumulates within the lysosome in presence of baphyllomycin in cells treated with FGF18. And this was also true in vivo. This is a skeletal element of a Medaka fish that was grown in presence of FGF18. And as you can see, there is more colocalization between LAMP1, which is a marker of autophagosome, of lysosome, with PDI, which is a component of endoplasmic reticulum. So suggesting that FGF18 is both in vitro and in vivo bona fide regulator of ER phagy. And this was also, these data were also supported by electron microscopy analysis. So we then searched for a possible mechanism by which ER phagy was regulated by FGF18, and we did phosphoproteomic analysis, and we, in collaboration with Matthias Mann group, and we found that basically FGF signaling in chondrocyte was indeed inhibiting the PI3, the insulin signaling PI3 kinase pathway. So basically we did also some molecular mechanism. So we found that FGF18 by activating JNK induced the phosphorylation of IRS1, and this phosphorylation is a signal for IRS1 to be degraded. So IRS1 is a major intracellular regulator of the insulin signaling, and so by degrading IRS1, basically the AKT-mTOR signaling pathway was inhibited by FGF18. So we know that the PI3 kinase mTOR signaling pathway is an important regulator of the activity of TFEB transcription factor. This transcription factor was originally identified by Marco Sardiello, which will be one of the next speaker as a postdoc in Balabiol's lab. And this transcription factor, once in the nucleus, activated the transcription of several genes involved in lysosomal biogenesis and autophagy. So this is considered a master transcriptional regulator of cellular catabolism. So we found that FGF18 is actually promoting TFEB activation. As you can appreciate that TFEB and its parallel TFE3 actually are normally located within the cytoplasm, but then after FGF18 translocate within the nucleus. And this was dependent on the receptor, FGF receptor 3 and receptor 4. And here you have a quantification of both TFE3 and TFEB nuclear translocation upon FGF18. So of course, this led to the upregulation of several lysosomal and autophagy genes upon FGF18. So I hope you can appreciate this heat map. So red means more expressed. And so as you can see, FGF18 induce significantly the expression of lysosomal and autophagy genes. And as a consequence, you can actually appreciate the fact that there is a strong increase in lysosomal biogenesis in chondrocyte treated with FGF18. But what was more striking for us is that FGF18 was indeed strongly inducing the expression of the ER phagy protein FAM134B. So as you can see, FAM134B by protein level and at transcriptional level is strongly induced by FGF18. And this induction is dependent on TFEB and TFE3 activation because, as you can see, is almost completely blunted in chondrocyte lacking TFEB and TFE3. And we think that FAM134B is a direct TFEB target because it's actually, we could find a TFEB binding site in the promoter of FAM134B, and we validated that by chip analysis and by luciferase assay that indeed the TFEB is promoting FAM134B through direct binding to this promotorial region. as a consequence, we demonstrated that in cells lacking T-Fab and TFE3, FGF18, but also nutrient starvation, the activation of Phagy is severely impaired in chondrocyte lacking T-Fab and TFE3. So further demonstrating that these transcription factors are indeed involved in the activation of Phagy, both upon FGF stimulation, but also upon nutrient starvation. So, and this is the data showing that during nutrient starvation, which we know is a potent inducer of autophagy, the activation of Phagy indeed require both FAM134B, because in cell lacking a functional domain of FAM134B, you do not have accumulation of fragments within the lysosome, and you also require T-Fab and TFE3, because you do not have, also in this case, in cell lacking T-Fab and TFE3, you do not have a significant accumulation of Phagy or fragment within the lysosome as observed in starved control cells. So the conclusion of this data that I show you is that in chondrocyte, at least in chondrocyte, the FGF signaling, but as well as nutrient starvation, by inhibiting the PI3 kinase mTOR signaling pathway, are actually activating T-Fab and TFE3. And then once in the nucleus, T-Fab and TFE3 not only promote biogenesis of new lysosome and autophagosome, but also induce the specific delivery of fragment within this newly forming autophagosome, and then the degradation of within lysosome. So basically, this is one of the first reported mechanisms of how during bulk-induced autophagy, some sort of selectivity in terms of substrates that will be recognized by the autophagy machinery exists. And we think that this selectivity is given through the transcription factor T-Fab and TFE3 that controls the expression level of FAM134B. So basically, we know that autophagy dysfunction is actually implicated in several human disorders, such as disorder of central nervous system, such as Alzheimer's disease, Parkinson's disease, and others. So it's involved also in cardiomyopathy, in different diseases of liver, such as alpha-1 antitrypsin deficiency. And also autophagy dysfunction, we and others demonstrated that autophagy dysfunction is also implicated in the pathogenesis of lysosomal storage disorder, or several lysosomal storage disorders. In particular, we and others have demonstrated that in cells affected by lysosomal storage disorders, autophagosome can properly form, sequester material, but then this material, this autophagosome cannot efficiently fuse with the lysosome and deliver autophagy cargo to the lysosome. In addition to that, autophagy cargo, the autophagy cargo that are inefficiently but still delivered to the lysosome start to accumulate within the lysosome because of a defective, of a global lysosomal degradative dysfunction. So basically, in this way, you have, we and others demonstrated that there is a defective autophagy flux in lysosomal storage disorders. So there are several experiments that have been done in this context. So I'm just showing you one as an example. So you can, in order to study autophagy, you can express in the cells an autophagy marker, an autophagosome marker, which is the LC3 protein, fused with the GFP and RFP tags. And then, as you can imagine, this protein is appear normally yellow because there is a colocalization between the RFP and the GFP tags. However, once this protein is delivered within the lysosome, then the GFP is quenched by the low pH of the lysosome, and so only the RFP fluorescence will remain. And so in this way, you can actually, by quantifying the ratio between yellow to red-only dots, you can actually quantify what is so-called the autophagy flux. So how much of the autophagy substrate are delivered to the lysosome and degraded by the lysosome. And as you can see in MPS 7 knockout chondrocyte, you can actually see that almost almost all the dots are yellow, so suggesting that autophagy are not delivered, autophagosome are not delivered, autophagy proteins are not delivered to the lysosome, and within the lysosome are not degraded. While in control cells, you can actually appreciate that the majority of the cells are of the spots are indeed red. And this is because the fusion efficiency between autophagosome and lysosome in normal cells is very high. So now the question is, we know that there is an impairment of autophagy in lysosomal storage disorder. So the question my lab would like to address is whether a selective autophagy dysfunction play any role in lysosomal storage disorder. So whether an impairment of specific substrates that are normally targeted to the lysosome for degradation through a selective autophagy mechanism, whether the accumulation of some specific target can actually account for some phenotypic manifestation in lysosomal storage disorder. So this is really what we want to address in the next year in my lab, and this project has been actually funded by the MPS Society. So we started from an observation that was done in another lab. So it was published in Journal of Anatomy last year. So as you can see, those are chondrocytes from MPS 1 and MPS 7 dogs. So as you can see, in this chondrocyte, one of the most striking cellular phenotype was huge ER dysfunction. So as you can see, those are in normal DR is very small. DR lumen is very small, but as you can see here, you have a huge distension of ER lumen in this chondrocyte. So this was really remembering us what we were observing in chondrocyte from mouse model that we generated lacking autophagy. And these data were really supporting our observation, published and unpublished observation, in which primary chondrocyte isolated from MPS 7 mice do have an accumulation of intracellular collagen. Similar to what we have observed in cells lacking Phagy. So we do have, as you can see here in green, you have collagen that form these dots, that we know these dots are located within the lumen of the endoplasmic reticulum. So here we have MPS 1 osteoblast, and I hope you can appreciate that they do accumulate procollagen. And again, in primary chondrocyte from MPS 7 mice, we do see actually that the trafficking of collagen is actually impaired in this chondrocyte. And also, if you perform RNA-seq analysis in this primary chondrocyte, you can actually see that the expression level of several of collagen genes are indeed impaired. So taken together, this observation suggests that in lysosomal storage disorder chondrocyte, you have an enlarged DR and accumulation of collagen. So this really is suggesting you that there might be an impairment of Phagy. So in order to do that, in order to study selective autophagy, and in particular Phagy in MPS 7 cells, we have developed a tool that can be used both in vitro. I mean, this was in vitro was developed by others, but now we have implemented this approach in order to expand also to in vivo this study. And we basically generated an ER protein, but as well as mitochondrial protein and lysosomal protein that is fused with GFP and RFP. And so in this way, once these ER fragments are delivered to the lysosome, then these ER fragments are not anymore yellow, but instead they appear only red. So by quantifying the red puncta, you can actually appreciate how much of Phagy there is in these cells, how much of mitophagy there is in these cells, and how much of lysophagy there is in these cells. But also we have basically cloned this construct in an AAV virus, and this AAV virus encodes for MRFP, GFP, ER proteins, and this is an ER sequence, the key del sequence. So as you can see, mice in hepatocyte on infected mice feed ad libitum, this protein localized in the endoplasmic reticulum, as you can see by the classical shape of the endoplasmic reticulum. But if you fast these mice, then these red dots appears, and these are bona fide ER fragments that are delivered to the lysosome, and then the GFP is quenched, and so you have the appearance of this red dot. However, if you repeat the same experiment in mouse in which autophagy cannot be induced due to genetic mutation in the mTOR signaling, so in these mice, even if you fast these mice, there is no induction of autophagy, then you clearly see that there is an impairment of the Phagy. So suggesting that actually this system is working quite well in vivo. And so I'm just showing you preliminary data. So we infected this adenovirus, we infected wild type and MPS 7 knockout mice. And as you can see, this is an infected hepatocyte from a wild type starved mice. And as you can see, you clearly see that there is appearance of red dots because, of course, starvation is inducing the Phagy. And so this can be quantified by the appearance of red dot. However, as you can see, GAS-B knockout mice has no almost undetectable number of red dots, suggesting that indeed these cells have defective Phagy. So what we want to do now in the next future is first to understand which kind of selective autophagy are defective in different tissue of the MPS mice. And then in parallel, we are doing high content screening analysis by using similar probes to identify existing drug and also new drugs that are bona fide inducers of Phagy. And so this is just a preliminary data. So as you can see, of course, it was gratifying that our high content screening picked up several inhibitors of the PI3 kinase mTOR signaling pathway, which we already known are potent inducer of Phagy, but we also found different additional molecules that may work through independent pathway and in the induction of Phagy. And so our idea will be to test these compounds in mouse model of MPS and then evaluate whether we could rescue the Phagy defects in these mice. And we hope that this might actually work in parallel with enzyme replacement therapy or gene therapy in order to ameliorate the specific aspect of the phenotype that are somehow resistant to classical therapeutic approach that are currently being used in lysosomal storage disorders. So in conclusion, and of course here I include also future perspective, Phagy is dysfunctional in lysosomal storage disorder cells, and an impaired homeostasis might contribute to the pathogenesis of LSD. We have already very convincing data that this is actually at least important for the bone phenotype, because we know that in bone-forming cells and cartilage cells, such as chondrocyte and osteoblast, we have an accumulation of collagen and an impaired collagen secretion, which might contribute to the skeletal phenotype. And so I show you some tools demonstrating that selective autophagy can be studied both in vitro and in vivo using fluorescent reporters. And as a future plans, we plan to identify new pharmacological modulators of Phagy and test them in lysosomal storage disorder models. So finally, of course, I want to acknowledge people that in my lab that perform most of the work that I show you, in particular Laura Cinque, Chiara De Leonibus, and Francesco Paolo Iavarone. and then former lab members such as Alison Forrester and Rosa Bartolomeo, different collaborators, and in particular also financial support. I again want to acknowledge the National MPS Society, which is actually supporting my work in the next year on selective autophagy and lysosomal storage disorder. And I will be happy to answer your question. Thanks.
Thank you, Carmine, for the great presentation. Really interesting. I have a question, which is really related to, I have a couple of questions. The first one is, you mostly studied the ER phagy in the context of collagen, but then you imply that other tissues and perhaps there's other pathways that are implicated. I wonder if you could maybe talk about it for a second.
Yeah, so of course we ended up with collagen because we were working in skeleton, and so skeletal cells, of course, produce huge amount of collagen, and so we found that this was accumulating in the R. But this is, I think, also due to the fact that collagen is a non-arid substrate. So basically, collagen is a very, once it's formed as a trimer within the endoplasmic reticulum, if it is misfolded, cannot be handled by the classical arid pathway, so cannot be retrotranslocated within the cytoplasm and degraded by the proteasome. So we think that in this particular condition, ER phagy is important because it's actually removing collagen and other arid-resistant clients from the lumen of the endoplasmic reticulum. And so we described collagen, but then Maurizio Molinari's lab described alpha-1 antitrypsin. Then there was another lab in liver that described ApoB proteins. So of course, I think if I have to imagine, we have to consider two parameters in order to understand which are the possible clients. So the amount of protein that is being produced by a specific cell type and the nature of the protein. So the nature of the protein, I think, has to be in a way that cannot be a classical arid pathway. So it's a more complex biophysical, from a biophysical point of view, has to be a more complex protein, and so needs to be handled by the ER phagy.
And another question I had is it's about the possible therapeutic use of these findings. And so if I understand correctly, it would be possible, I mean, you're exploring pharmacological modulation, of course, but then would it be possible to simply overexpress 134B as it was attempted with, done with TFEB?
Yeah, so of course also the overexpression of TFEB worked very well in mice, but I don't think there will be any, at least in the near future, any gene therapy using TFEB due to the side effect. So, and I think for FAM134B, there might be similar problems in the sense that this, so I don't think it's now safe to imagine that overexpressing a protein that has a reticulum homology domain in the R that may cause large ER fragmentation, and the ER phagy can be finely tuned within the cells. So I think we should be very careful in planning that. So of course, using already approved drugs that we know are inducing autophagy, however, from a safety concern, we already know that they are well tolerated, and so. But of course, having said that, I think it's at least in preclinical animal, it should be it should be tested, the viral mediated gene delivery of ER receptors. The other point that you should consider is whether once delivered within the cells, this ER Phagy receptor needs a stimulus to be activated. And so this is also another aspect that we are working on. So we found that some of them are constitutively active. So as soon as you overexpress them, they induce ER Phagy. Other will require a post-translational modification such as dephosphorylation. And so maybe this could be a safest way to express an inactive protein and then to pharmacologically activate it.
Yeah, there is only, exactly, it's only when it's the right conditions are there. I'm looking for the chat and I was wondering also, I'm trying to go back. So, and there's no pathology associated with mutations in
Phagy receptor. They are mostly sensory neuropathies.
Sensory neuropathies.
Sensory neuropathies. So the mechanism is unknown, but it's possible that basically these neurons that have very long axons may require remodeling during development. But of course, there is no formal demonstration that this is the mutation in these genes are actually causing the phenotype because of an impairment of ER Phagy. So, of course, one can speculate that these proteins not only works in ER Phagy, but they also may have additional function in ER shape remodeling, and so it will be might be very well possible that the phenotype is due to the lack of another function. But it's peculiar that in at least three or four ER Phagy receptor out of 8 are associated to sensory neuropathies in human.
Okay, great. I think we're at time. I want to thank you again for the really wonderful talk.
Thank you so much.
Very interesting. And so we're moving on to the next speaker now, Elena Pavlova from the University of Cambridge in UK. So she's going, the title of her presentation is MPS+, a lysosomal disease caused by mutations in VPS33A and impaired lysosome dynamics. Elena?
Yes, thank you. Good afternoon. I just would like to share our experience and studies where we discovered a new disease, which is now called MPS+, and has a mucopolysaccharidosis phenotype. So we studied patients from Yakutia, which this is a place located in far east of Siberia. So there were several patients with a similar phenotype, which were referred to us internationally from National Hospital of Republic of Sakha-Yakutia. So we studied 3 children from 3 unrelated families with severe disease that has a particular similarity to mucopolysaccharidosis. And with the estimated frequency in Yakut population is about 1 in 14,000 live births. So the disease affected ethnically Yakut infants. Typically by the age 6-9 months, children have developmental delay. They have diffuse hypertonia, restricted joint movements. Also, it was found that they have a delayed myelination, delayed speech development, and often they can't stand without support. They have a cerebellar atrophy and brain atrophy. Unfortunately, the children died up to age 3-4 years old. So the main clinical characteristics include MPS-like clinical signs, including coarse face, short neck, skeletal dysostosis, and characteristics hands with like claw-shaped hands, hepatomegaly, usually associated with recurrent pneumonia. In addition to that MPS-like clinical signs, patients also develop systemic complications, including thrombocytopenia, anemia, neutropenia, kidney disease, cardiomyopathy. So the kidney disease actually started with a nephrotic syndrome, which we call when the patient developed quite severe proteinuria, hyperalbuminemia, and associated with that marked peripheral edema. On autopsy examination, it was found that the kidney was with glomerulosclerosis. And you can see on this slide they were found vacuolated podocytes. So the next step in our diagnostical screenings, diagnostic research, was to test urine glucosaminoglycans in urine as in a normal clinical settings with the patients who would develop MPS-like signs. And you can see on these slides, we found that there was an accumulation of specifically dermatan and heparan sulfates in all 3 patients. That would suggest that the patients may have MPS 1, 2, 6, or 7, but all related lysosomal enzymes actually were normal. And then that was confirmed in 3 laboratories in Moscow, Manchester, and London. So then to summarize the clinical lysosomal screening, we found normal lysosomal enzyme activities in all patients, accumulation of dermatan and heparan sulfates, slight increase plasma hexosaminidase A and B and monosidase, and also importantly we saw increased of glycoconjugates in urine with normal free sialic acids and normal activity of lysosomal neuraminidase. Then on the next step, we tested, we done Lysoplex panel for 89 lysosomal genes in collaboration. So it was done in collaboration with Dr. Balabio and Teleton Institute in Italy. And also we conducted a next generation sequencing for on another panel using 50 lysosomal genes, and no significant variants were found. So we excluded all known lysosomal diseases, including many MPSs like MPS 1, 2, 6, 7, mucolipidosis type 2 and 3, MPS 3 type C, B were also excluded. So on the next stage, we conducted a next generation exome sequencing in 1 family, 1 patient and 2 parents, and 2 other patients. We knew that in Yakut population, there is a high risk of autosomal recessive diseases, and we applied autosomal recessive model in filtering of exome sequencing data and found a homozygous missense mutation in conserved region of the VPS33A vacuolar protein sorting associated protein 33A. And this mutation predicts replacement of arginine by tryptophan at position 498. And Sanger sequencing confirms segregation of this mutation as a Mendelian autosomal recessive trait, as you can see on this slide. And the mutation was found in all 3 patients. So we also found that there was described 2 patients, 2 Turkish patients with the same mutation and the same phenotype. You can see it's a mucopolysaccharidosis-like signs. And in addition to them, there were systemic complications. And unfortunately, the patients also died in early childhood. So the VPS33A is part of Hope's complex, homotypic fusion and vacuole protein sorting complex that was first identified in genetic screens of yeast. But it's actually conserved across eukaryotes and forms large multi-subunits shown here. So the Hope's function is to accelerate vesicle fusion, and it forms vesicle tether between vesicles and regulate rate of snare-mediated fusion, catalyzes snare complex assembly, and accelerates snare-mediated fusion. So we also know that there is a buff mouse with another missense mutation. The mutation in mouse VPS33A located in a different domain than we found in our patients, in the protein, in the mutant protein in our patient. And the mice have motor dysfunction and cerebellar atrophy and loss of Purkinje cells, as you can see on this slide. So this is a normal mouse where you can see clearly the stained, these dots stained for Purkinje cells marker. And in the buff mouse, it's quite significantly reduced and caused cerebellar atrophy. So the mouse also showed decreased platelet activity, resembling, as they thought, Hermansky-Pulbach syndrome, a bleeding diathesis with hyperpigmentation. The mouse also showed in the experiment impaired autosomal autophagosome lysosome fusion. So mice showed defective motor learning on the behavior test, and also developed difficulties in motor function at older age. So how this missense mutation in VPS33A impairs lysosomal recycling of glycoconjugates is unclear. We can see that the accumulation of dermatan sulfate and heparan sulfate and glycoconjugates, but it would require, of course, the more detailed studies on functional relationship between the autophagosome, lysosome, and endosomal compartment, and the dynamic exchange of the contents between them. So, but I would like to say that patients with suspected MPS with specific urinary GAG and oligosaccharide pattern, as we can see in these patients, and normal lysosomal enzyme activities should be considered for diagnosis of MPS plus syndrome and screened for genetic defects in the VPS33A gene. So we next to come back to our studies, we wanted to see how that mutation affects the protein function. We modeled the mutation in the structure of VPS33A. And as you can see, residue arginine 498 form salt bridge with aspartic acid 484, and also interacts with with other network, with other domain, with other amino acids. So replacement of arginine 498 to a hydrophobic residue tryptophan is likely to disrupt and loosen the association between domains. Sorry, why? And between domains 2 and 3b. The location in domain 2 of VPS33A does not correspond directly to the binding interface of with VPS16, which has been extensively studied by our colleagues in Cambridge, and that predicted but we predicted that this mutation can destabilize the folded structure of the protein as a consequence to impaired assembly and stability of the Hobbs complex for function, fusion of endosomes with lysosomes, and possibly another complex corvette, which called corvette, which work with at earliest endocytic pathways. So when in vitro experiment involving knockout of human VPS33A in HeLa cells have recently been reported by our colleagues in Cambridge, and they showed that the protein is required for endosome and autophagosome Hobbs-dependent membrane fusion with lysosome. It was identified that complex of VPS33A and VPS16 particularly is highly important in membrane fusion lysosome with endosomes and autophagosomes. And Hobbs complex forms physical tether between vacuoles and mediates snare-mediated fusion. So this is an important function of Hobbs complex, which accelerates snare-mediated vesicle fusion. So we wanted to check, we cultured patient fibroblast and wanted to see if our prediction was instability of the VPS33A and assembly of the Hobbs complex would be impaired. We conducted this immunoblot experiment. And as you can see, there was a reduction of VPS33A in both patients. And also, it's important to see that the other proteins in this complex, I'm not sure why it's, other proteins in this Hobbs complex also been reduced. So we predict that deficiency of the VPS33A leads to impaired assembly and deficiency of the Hobbs complex and potentially another corvette complex. So when we looked on the cells on electron microscopy, we found that there was increased vacuolation and expansion of endosomal lysosomal compartment in patient cells. As you can see, that the morphological changes in the cell with this mutation. And it may be due to imbalance between fusion and fission event can result in swollen endosomes and lysosomes looking like that. So we also and others found that there was an increased acidification of the lysosomes. And that could lead to swollen vacuolar compartment. As you can see on these slides, we stained cells with lysotracker and lysosomal pH sensor, and there is a high bright signal in patient cells in comparison with the control fibroblasts. So the explanation of this could be that the proportion of acidic swollen endolysosomes due to deficiency of Hobbes complex, causing an overall reduction of mean lysosomal pH and alteration of the pH profile. So in these cells. So to investigate further the defect of the mutation on BPS33A, we stably expressed mutant protein on human BPS33A in HeLa cells and compared it to wild type expressed protein. And we used a proteasomal inhibitor called MGS132 to see if this would change the abundance of the mutant BPS33A. And it's indeed, as you can see on this slide, quantification of data from 4 experiments showed that after exposure to the proteasomal inhibitor, for 6 hours, the abundance of VPS33A increased in the cells expressing mutant VPS33A. So then I would like to explain maybe a little bit more on the potential trafficking defect which might be caused by the deficiency of this Hobbes complex and VPS33A. So we know that in normal cell, the delivery of endocytosed macromolecules, including glycosphingolipids, to lysosomal hydrolysis requires series of fusion and fission events between late endosomes and lysosomes, also known as multivesicular bodies. So, and where this degradation would and recycling of macromolecules would occur. But, it is possible that if deficiency of the Hobbes complex it could delay endocytic delivery to the of the substrate via insufficient fusion events and resulted in accumulation of these substrates in lysosomes, and including accumulation of glycosphingolipids. So in fact, this phenomenon has previously been observed, and several unrelated sphingolipid storage diseases have common sphingolipid trafficking defect associated with lysosomal accumulation of sphingolipid, but caused by different primary abnormalities. and it was observed in GM1, GM2 gangliosidosis, metachromatic leukodystrophy, Niemann-Pick type A and C, Fabry disease, and Krabbe. So on this slide, you can see that in the experiment with the fluorescent lactosyl ceramide, when it's endocytosed in the cell, in normal cell, it would be delivered to perinuclear area, to Golgi, here. But in the disease, we see that image. where there is fluorescent signal found in punctate structures. So it's delayed, the trafficking defect would be shown like this in the cell. So we, to explore why the sphingolipid trafficking may be affected by the reduction of VPS33A, we carried out fluorescent, the same experiment with fluorescent lactosyl ceramide labeling and chase experiment in the patient and control fibroblast. And endoscytosis lactosyl ceramide in normal cell after 3 hours of text was delivered to Golgi, as you can see on this image, in normal fibroblasts. But in the patient's cells, we observed the same phenomenon as in other sphingolipid storage diseases with peripheral punctate structures. Consisted this accumulation of the substrate in lysosomes, in lysosomes or endosomes. But we wanted to check if this our protein involved in this process, and we treated cells with a proteasomal inhibitor to stabilize mutant VPS33A, as we confirmed on Western blot, and by looking at the same delivery of fluorescent lactosyl ceramide, we can confirm that after incubations with lactosyl ceramide, we observed only signal in the perinuclear area, so that in the Golgi complex. So that's also we analyzed normal glycosphingolipid recycling by analysis of concentration of glycosphingolipids in the cells, and we found that in one patient there was increase of glucosyl ceramide and galactosyl sphingosine. But we tested only 2 patients, but another patient, the cells was about a normal concentration. So we also we further stained cells with philippine, a fluorescent marker that specifically binds to the cholesterol. It is important to say that in fibroblasts of patients, philippine fluorescence was increased, suggesting increased levels of cholesterol, as found, for example, in fibroblasts from patients with other sphingolipid storage diseases, as well as Niemann-Pick type C. So we quantified this punctate after treatment with bortezomib and showed a greatly reduced number of the peripheral puncta and bortezomib treated patient cells. This suggests that a proteasomal inhibitor, such as bortezomib, actually it's licensed for treatment of multiple myeloma currently, could be explored for potential treatment for patients with this mutation in VPS33A. So next, we also studied inhibitors of glucosyl transferase that catalyzes the first step in glycosphingolipid biosynthesis, synthesis of glucosyl ceramide, which have been administered to patients with several sphingolipid lysosomal storage diseases as part of substrate reduction therapy. To explain how it works, just in the presence of lysosomal dysfunction caused, for example, by deficiency of glucosyl ceramidase, too much undegraded glycosphingolipids are not recycled properly. In the situations of complex deficiency or other diseases with delayed trafficking defect, the delayed trafficking and subsequent recycling of glycosphingolipid is impaired, leading to secondary storage of the glycolipids. And this therapy is designed to reduce its de novo synthesis, and it does so by ceramide analog, which is highly specific inhibitor of the de novo synthesis, and it's possible to reduce buildup of glycosphingolipids. So we conducted a next experiment. We treated patients' cells with clinically approved drug inhibitor, eliglustat, which is currently licensed for treatment of Gaucher disease. And data from 3 independent experiments showed an improvement in lactosyl ceramide trafficking in the cells from the 2 patients. And after incubation with eliglustat, as measured by a reduction in lactosyl ceramide labeled puncta, this suggests that the treatment with eliglustat could be further investigated for use in patients with MPS plus disease. And finally, I'd like to say that recently 2 patients with mutation in the core protein of Hobbs complex and another protein VPS16 were described. The intronic mutation in VPS16 disrupt normal splicing, and it's been shown that it caused deficiency of VPS16 as well as VPS33A, and as well as reduction of the Hobbs complex. These patients have the same very similar phenotype with patients from Yakutia with MPS-like signs, and the characteristic accumulation of dermatan and heparan sulfates, as well as increased glycoconjugates. So in summary, I would like to say that MPS resembling disease found in 18 patients of Turkic ethnic groups with homozygous mutation in VPS33A, reduction of VPS33A, VPS18, and 41 protein suggests a reduction of assembled Hobbs complex and defective membrane recycling via endosome-lysosome system. The mutation in VPS33A induces a clinical phenotype of a lysosomal disease due to a novel pathological mechanism that is regulated by Hobbs complex. Structural defects in the Hobbs complex and VPS33A protein has profound effect on the morphology and function of endosomal lysosomal compartment, including defective glycosphingolipid traffic, and stabilizing of mutant VPS33A with a proteasomal inhibitor, and as well as inhibition of glycosphingolipid synthesis suggests therapeutic strategies based on repurposing of clinically approved drugs. So, and finally, I would like again to repeat that patients with suspected MPS with specific urinary GAC and oligosaccharide pattern, where there is a normal activities of known lysosomal enzyme activities, should be considered for diagnosis of MPS plus syndrome and screened for genetic defects in these 2 genes now, VPS33A and VPS16. And finally, I just would like to thank you all my colleagues in Cambridge and our international collaborators who helped to find the cause of this disease in Yakutian patients and to help us with this study and studies of that protein. Thank you very much.
Thank you, Elena, and very interesting presentation. We don't have a lot of time, and but I also want to invite the audience to put questions through the chat if they have any. Maybe I can ask you one question. You mentioned some potential therapeutic approach. I was wondering if there's any planned trial ongoing or ongoing trial, sorry, using these approaches. And of course, these are only going to work for the specific mutation you presented in the first part of your talk, right? It is particularly the proteasome inhibitor. So if you can comment on that.
Yes, of course. Thank you. We certainly were thinking about clinical trials as a possibility to test, to try to use these treatment approaches in patients. But of course, it's a very early stages to say that we can move on to that stage. We would need to do more work to probably to confirm our finding in other cells, in more patients' cells, to have be more confident in these approaches. But having said that, I also wanted to say that there are other patients which we investigating at the moment who have mutations in the same protein, but in different location. So I think in the coming years, maybe we will conceive that as we have many undiagnosed patients where we couldn't find the reason and cause, that in those groups could be identified this disease, and that we may have a more broader, more bigger picture of the disease and how the treatment could be used to help them.
Great, thank you. Thank you again for the great presentation. And then we're gonna move to the last speaker of this session, Marco Sardiello from Washington University in St. Louis. And the title of the presentation is a stepwise mechanism for ER2 Golgi transport of lysosomal enzyme. Marco.
Good morning and good afternoon, everyone. And I would like to thank the organizer for the kind invitation. So today I will be showing some recent data from a lab, some results that are focused on the study of the trafficking of lysosomal enzymes. So we are familiar with, okay. It's not working for me.
It seems to be moving now.
Oh, okay, yes. Okay, so we are familiar with the concept of synthesis of lysosomal enzymes that happens in the endoplasmic reticulum. And sorry, well, it's just going slow on my side. Okay, all right, try again. Okay, yes, now I can see it. So enzymes are synthesized in the lumen of the endoplasmic reticulum, and they accumulate to ER excises where then they travel to the Golgi complex through COP2 associated vesicles. In the Golgi complex, they undergo transformation to display mannose phosphate tags, and this is the specific mechanism for their final delivery to the endolysosomal system. And so what was not known, okay, yep, is how the first step of the trafficking process happens. So we know that the mannose phosphate receptors are helping in the trafficking from the Golgi, the trans-Golgi network to the endolysosomal system. But how this first step of the trafficking was actually unknown. Do you see the screen going?
You have, it seems to be okay.
Yeah, it is just very slow on my side, so I never know when it goes on the next slide.
I see.
Okay, I'll try to go back.
I think that's the one.
Okay, sorry for that. So we hypothesized that there must be, yeah, you see, it bounces back and forth. Maybe 300 mega of file was not easy to manage. Okay.
Far away.
So we hypothesized that a specific ER cargo receptor is involved in the anterogate lysosomal cargo trafficking from the endoplasmic reticulum. And to identify candidates, and again, it's not moving. Okay, so to identify candidates, we started from a kind of transcriptomic approach. So we had developed a tool to investigate the co-expression profiles of any genes of interest. And so, for example, from previous studies, we knew that lysosomal genes are co-regulated with each other. This is a heat map showing the degree of co-regulation of lysosomal genes. So we basically reasoned that if there is any ER receptor that is serving the trafficking of lysosomal enzymes, maybe chances are that these ER receptors are also co-regulated with the lysosomal enzymes. So basically, we ran an analysis of all the about 800 genes encoding for resident ER proteins to look for genes that are co-regulated with lysosomal genes. And we found that about 10% of genes encoding for ER proteins are co-regulated with lysosomal genes. And this 10%, so we are talking of about 70, 80 genes. And among this pool, there were 4 that were actually, the proteins had a structure and possible role for which we could hypothesize that one of them could be the ER transporter for lysosomal enzymes. And these are TMET9, LMF1, TMET4, and Serenade. So we ran a screen of these proteins. We cloned the 4 of them, and we also cloned all the 50 or so lysosomal enzymes using the bimolecular fluorescent complementation system. So basically, in this system, you attach half of the YFP or GFP protein to one of your candidate genes, and the other half to the other gene. This is a system to probe physical interaction. And you get fluorescence in the cell when you transfer these 2 constructs. So you get fluorescence only when your proteins are interacting with each other. So we did the screen for basically all the hydrolases and these 4 candidate receptors. And if it goes to the next slide. Yep. Yeah, and we found that only one of these 4 candidates, which is Serenade, was actually able to interact with all the pool of hydrolases that we tested. You see, all of these green dots means that there is interaction between the pools of hydrolases and these particular proteins, whereas the other 3 really didn't give any noticeable signal. So we focused on selenate, and this time we tested the pairwise interaction between selenate and all the lysosomal enzymes. And so here we measure the intensity of the interaction by using, again, the bimolecular fluorescent complementation assay, but a flow cytometry analysis to measure the extent of this interaction. And as you can see, about 2/3 of the hydrolases of the lysosomal enzymes are significantly interacting with selenate. And this was also confirmed independently by co-immunoprecipitation assays. So here we are selecting just a pool of these enzymes and the negative control. So we have XB as a negative control. XB is not giving a signal in the fluorescence assay, and also does not co-immunoprecipitate with selenate. But all of our test proteins are co-immunoprecipitating with selenate. So there is physical interaction. Okay, so what is selenate? Selenate is an interesting protein. That's an interesting gene because even though it is an ER protein, it is mutated in a lysosomal storage disorder-like disease. So selenate disease is a neurodegenerative disorder associated with lysosomal impairment. There is accumulation of autofluorescent steroid material in most cell types. And what was nice in a sort of way is that the molecular pathology was completely unknown. Again, it looks like a lysosomal storage disorders, but selenate resides at the ER. So why there is this connection? So we first tested if loss of selenate is causing differences in the lysosomal proteome. So we have a mouse model, which is a natural mouse model for selenate disease. There is a friendship mutation in in the gene, so there is total absence of the protein. So we isolated lysosomes from this knockout mouse for selenate. Isolated lysosomes, and we performed proteomics analysis to analyze the content of lysosomes. And this is just showing the enrichment for lysosomes as measured by LAMP1, which is a lysosomal membrane protein, often used as a marker, and the enzymatic activity of XA, which again was not interacting with selenate. So in our lysosomal enriched fraction, we perform proteomics analysis. And if it goes to the next slide, yes. And we saw that basically all of the hydrolases were downregulated, so their levels were lower in the knockout mouse. So everything you see under the line here of 1 is depleted in the knockout samples compared to the wild type samples. And but this was not happening for the lysosomal membrane proteins, the red dots here. They actually, as you can see, they're not really depleted, or if anything, they are slightly enriched, at least some of them. And more in general, so when we put all of this data in a single graph, and we also use, for example, mitochondrial protein, which were contaminated in our samples, just as an external control, we see that this depletion is really specific for the lysosomal soluble components. Lysosomal proteins are fine, equally distributed in the wild type and knockout, mitochondrial protein equally distributed in the wild type and knockout. So this is really a specific depletion for the lysosomal soluble proteins, which are the enzymes. And this was also confirmed by Western blot analysis in our samples. So again, you can see there is a deep depletion in several hydrolases that we tested, but not of LAMP1 protein, for example, here, that we use as a positive marker. So next question. Oh, sorry, it's again very slow. So then we focused on the interaction of selenate with the enzymes. So in the next slide, I'm reporting the putative structure of selenate. As you can see, it is a multipass membrane protein, transmembrane protein. And there is a very large luminal loop. So this loop is protruding in the lumen of the ER. And we also performed some evolutionary conservation analysis and using selenate sequences from all available vertebrate species. And we found that this loop corresponds to the most evolutionary conserved region of the protein. So, well, we did the obvious hypothesis that maybe selenate was interacting with the enzymes by using this loop here. And so we also did the obvious experiment. We prepared the construct of selenate in which we cut this loop to see what would happen with the interaction. And so first of all, we verified that selenate with this delta loop is still chorally expressed and still chorally localizing in the R, which is the case. So the green signal is not really distinguishable from the red signal, which is the wild type selenate. So everything good here. The protein is stable, it is expressed, it goes to the ER. And it is also able to work in the bimolecular fluorescence complementation assay, because so selenate is a homodimer. And so here we are testing if selenate is with the delta loop, with this loop cut, could still be able to interact with itself, which is the case. So it is competent for this assay. But when we test the interaction of this construct with the hydrolases, as you can see, the interaction is completely gone. So the deletion of this loop disrupts the interaction of selenate with the lysosomal enzymes. So next, we examined a few pathogenic mutations that were previously mapped on this loop, and I have them here in the next graph. So as you can see, there were several mutations. Those are missense mutations that were mapped in the loop. So we prepared as many constructs, each of them carrying one of these mutations. So again, all of these constructs were able to self-interact with no problems. So their signal in the bimolecular fluorescent assay experiment was indistinguishable from the wild type. But interaction with several pools of hydrolases that we tested was really impacted by this mutation. So probably, in these patients, this is the mechanism of pathogenesis. So those mutations are impacting on the interaction of Serenade with the lysosomal enzymes. So next, we asked if Serenade has any partner proteins or if it is working just by itself. And in particular, we were attracted by the fact that Serenade is one of the neuronal steroid lipofuscinosis proteins, and which is a family of more than 10 diseases, as I have in the next slide. And most of these CLN proteins are lysosomal. There are several enzymes, membrane proteins, but there is another one which is called CLN6, which is also associated to the membrane of the endoplasmic reticulum. And it is the only one in this group of diseases, other than Serenade, is the only one that is also associated with the endoplasmic reticulum. And it's also interesting to observe that Serenade and CLN6 disease patients have really broadly overlapping phenotypes, and also some of the described cellular features previously associated with CLN6 and Serenade diseases have overlap. And this is a protein that, by the way, is not homologous to Serenade. These proteins are just named after the disease. There is no sequence homology at all. And this is the structure of Serenade. So first of all, we wanted to investigate if also CNN6 is involved in the pathogenesis of lysosomes. And so there is a mouse model for CNN6 disease as well. There's another natural model with another frame shift mutation in the gene. So we again isolated lysosomes and performed in this case immunoblots. And so again, there is a depletion, a deep depletion of lysosomal enzymes in the lumen of lysosomes in the knockout mice compared to the wild type. And here we are also showing some enzymatic activities. But again, not of membrane proteins, LAP1 is fine. Yes, sorry, in this slide, I had the enzymatic activity. And this is not due to transcriptional changes, because we performed real-time PCR to monitor the expression of these genes, and they were fine. If anything, they were even upregulated. So that's, again, something that is happening post-translational. So we wanted to see if there is any interaction between CLN6 and Serenade. At this point, the main question was, are they really partner, or maybe they are just serving a sort of redundant mechanism independently of each other? So we tested interaction between CLN6 and Serenade. And again, using first the biomolecular fluorescent complementation assay. And yes, we found interaction between these two proteins. And here we just have a negative control with LMF1, which was one of the initial candidate proteins. There is no interaction with this protein. And we verified the interaction between CLN6 and Serenade also by using, again, co-IP experiments. And we also used BIFC to map where this interaction was happening, and we confirmed that it was happening in the endoplasmic reticulum. Here we are using KDL as a marker for the endoplasmic reticulum. So all good so far. We wanted to understand if CLN6 trafficked to the Golgi complex. We knew already that Serenade was trafficking to the Golgi complex due to previous published literature data that I don't have the time to show, unfortunately. So we did some experiments specific for CLN6. And in particular, here we are focusing on a putative signal, AAA, which is a putative retention signal in DR for ER-associated proteins. So, because in normal immunofluorescence, CLN6 only overlaps with ER markers. So, as you can see here, we are using KDL but does not overlap with Golgi marker. So here it is KDEL, but it should be GM130, which is a Golgi marker. So there is no overlap at all with Golgi markers. So we mutated CLN6 to see if mutation of this retention signal would change that. But again, it is massively overlapping with the ER marker, but not with a Golgi marker. And then we also did a slightly more complicated experiment by using vesicle budding in collaboration with Randy Sheckman. And this is an in vitro assay that is based on the in vitro assembly of the COP2 associated vesicles that are the vesicles that physically allow the transportation of proteins from the ER to the Golgi complex. So here we are using several POSI controls that are known to be transported through COP2 vesicles, including, for example, APP and ERGIC 53, etc. And so when we mark Serenade with a MIC tag, we see that where all the controls are in place, Serenade is incorporated in these COP2 vesicles, but not CLN6. So we were never able in any condition to see CLN6 being incorporated into these vesicles. We also tested a lot of other systems that are in the papers that we have published. We really tried hard to try to make CLN6 go into the Golgi, but we were never able to make CLN6 go to the Golgi. So we concluded that it is not going to the Golgi. So next, we wanted to see if CLN6 is able to interact with the lysosomal enzymes. And these experiments were really straightforward. So we again performed co-immunoprecipitation. So these are very heavy immunoblots. So it's taking some time to switch slide. Okay. Yeah. And so these are co-IP experiments. And again, you can see that CLN6 is able to interact with lysosomal enzymes. And those are the same enzymes that also were interacting with Serenade. We also did the same experiments. So CLN6 also has a big loop that is present in the lumen of the R. So we made a construct in which we eliminated this loop, and we tested again the interaction. And as you can see, when we use the loop, it is the third line in each blot. When we use the delta loop construct, the interaction is basically gone with the enzymes. So that seems to be a very similar mechanism to Serenade. But then again, we wanted to know, okay, are they working together? Are they in a complex? So are they necessary to each other for this interaction and for this transportation of the lysosomal enzymes? So we used CRISPR editing to generate knockout cells for the CLN6 and Serenade. So here we are using CLN6 knockout cells to test the interaction of Serenade with the enzymes. So those are the wild-type cells. As you can see, there is interaction, the second line, with the usual enzymes that you are using for all of these tests. But when we do the same experiment in the absence of CLN6, the interaction is gone, the third line in each of these blots. And we also did the reverse experiment. So we tested if Serenade is indispensable, is required for the interaction of CLN6 and the enzymes? And the answer is again, yes. So in Serenade knockout cells, the interaction between CLN6 and the enzymes that you can see in the first lines in these blots is gone for all of these enzymes. So they seems to be working in a sort of a complex. And then we also did some experiments to monitor the trafficking, the impact on trafficking of enzymes in the absence of Serenade and CLN6. I'm not showing the ones for Serenade, again, for lack of time. I'm just showing the ones for CLN6, which are visually more straightforward to display. So here we are synchronizing the trafficking of the enzymes with a nice system that we acquired from another lab. So at time 0 is when the enzymes are released for their hook in the endoplasmic reticulum. And so those are wild-type cells. And you see that in a few minutes, they transfer from the ER to the Golgi. In 20 minutes, for example, this is cathepsin D. In 20 minutes, all of cathepsin D is gone to the Golgi complex. But this does not happen in the CNS6 knockout cell. So there is still some transfer, but this transfer is less efficient. And here there is all the quantification and the p-value, etc. And we tested this for several enzymes. So those are the experiments for cathepsin D. We have similar experiment for PPT1 with similar results. We also have a negative control. In this case, we used lysozyme C, which is a non-lysosomal enzyme, and the impact of lyso C to the Golgi was absolutely not impaired by the absence of CNS6. So again, the systems seem to be specific for lysosomal enzymes. And finally, we also did some tests in vivo by using the two mouse models for CNS6 and Serenade diseases that we had to see, to finally test in vivo if they work as a functional unit. So here I'm showing just one of these tests, which is the monitoring of their lifespan. So here, sorry, this didn't come out nicely, but here we have the lifespan, we are monitoring lifespan of the single deficient mice. So Seren6 is in red, Serenade is in green. And then we made a double knockout mouse to see if having both genes knocked out will modify lifespan. But this did not happen. As you can see, the double knockout has the same lifespan of the worst of the two models, which is Serenade. Of course, we were expecting if they were not working together, but in a sort of redundant way, then the double knockout will literally crash the lifespan of the mice. We performed similar experiment for vision loss, and that I'm not showing here today for lack of time, but they gave the same result. So the double knockout was similar to the two single knockouts. Okay. So we concluded that Seren6 and Serenade form an obligate complex for ER recruitment and ER to Golgi transfer of lysosomal enzymes. And we named this complex Aggress, which is ER to Golgi relaying of enzymes of the lysosomal system. This takes several hours of brainstorming and a couple of shots to come out with this acronym. And so we propose that, so we see that Seren6 and Serenade interact with the lysosomal enzymes via their large luminal loops. Serenade is recruited to COP2 vesicles via specific cytosolic signal that I didn't show, but it is in the papers that are referred here, whereas Seren6 is excluded from COP2 vesicles. And so loss of either component of the Aggress complex causes depletion of lysosomal enzymes at the lysosome. So, and here there is just a cartoon showing this mechanism. So Seren6 in red and Serenade in blue associate at the ER membrane, and they recruit enzymes for delivery to COP2 vesicles, which makes the transfer to the Golgi system. And in the absence of either Serenade or Seren6 or Serenade, this process is much less efficient because there is not a direct interaction of the enzymes with the components of this complex. And so there is a depletion of enzymes in the mature lysosomes. So the Aggress complex is the first ER cargo receptor system that is associated with the biogenesis of an organelle, and also is the ER cargo receptor with the largest set of client proteins known today in any mammalian system. So, and I would like to thank all the people who worked to these projects, and in particular, Alberto Di Ronza and Laksha Bajaj, who literally devoted years and years of their life to these projects, and really uncountable hours, if not years of work. And many collaborators and also all the foundations that support the work that we do. Thank you very much, and I will be happy to take any questions.
Thank you, Marco. Unfortunately, we're running horribly late and we have to close the session. I was told by the organizers, but I want to thank you all for the wonderful presentations, and then we are going to close here. I hope the audience had enjoyed the sessions as much as I did.
Sure. Thank you.
Thank you.
Thank you.